Hello and welcome to the Friendly Pharmacy 5 Book Club, Session 5. We have an incredible guest with us today who is going to educate us even further on all things gut health and respiratory infections and other common infections. So, if you have joined us in a previous session, let us know what your biggest takeaway has been from these sessions. But I'd like to welcome you, and if you can let us know in the comments maybe where you're from, or if you have any questions about gut health as we go throughout this session, please feel free to put those in the comments because we will be watching the chat section, and we will also be putting everyone who makes a comment or asks a question into a draw to win this book that we've been featuring, which is Gut Logic by Graham Mackenzie. And Graham Mackenzie is with us today, and I'm going to introduce him in just a moment. But first of all, I'd like to start with just a fun fact about the microbiome. So, did you know that identical twins have fairly distinct microbiomes? Though they share a lot of genetic information, their microbiomes are barely more similar to one another than the ones found in fraternal twins. This means that our microbiome is heavily influenced by our individual environment. And if you were with us in the first session, you will know, actually in the second session, where we talked about prebiotics versus probiotics. And we talked about all the different things that can affect the microbiome, like age, circadian rhythm, even travel, that kind of thing. I just thought that that would be a fun fact and a great way to start the session. So again, if you make a comment or ask a question, you may be the winner of a copy of Gut Logic by Graham Mackenzie. So we would love to interact with you in the chat. And so please feel free to do so as we go throughout this session. Without further ado, I would like to introduce the author of Gut Logic, who's been with us in all of these sessions. Graham Mackenzie is with us today. Hello, Graham. How are you doing?
Good. Hi again, Lindsay. It's good to be back again. We've had a number of really good sessions, and this looks like a really good one too.
Yeah, I'm really looking forward to our speaker today. And Graham, you were busy speaking this morning, weren't you?
Yeah, so I drove. Probably a lot of people on this call don't know a whole lot about Cape Breton geography, but I went up to Marguerite from Bedeck, about a 40-minute beautiful drive, and spoke to a group of seniors there. And we had a lot of great questions. And as we spoke off air, speaking to seniors is really it's a great way to get out of the pharmacy and just get a lot of really good questions.
Yeah, I think that they're one of my favorite audiences is when we go and speak to seniors groups because they're just so interested and engaged, right? Also,
Yeah.
for those who are just joining us, if you do want transcripts from our Q&A sessions, all of these sessions, we've been doing a Q&A at the end after the speaker presentation. So if you would like Q&A transcripts, you can sign up for our newsletter at friendlypharmacy5.com, and that will give you access to the transcripts. The newsletter is also a place where I share things that I can't necessarily make a video out of, different insights on health or different news on health, right? And all of this is evidence-based. And so if that's something that interests you, you can go to friendlypharmacy5.com. So I think that that is about it. I'm going to introduce to you the speaker today. So we are very honored to have Dr. Paul Wischmeyer with us today. So let me tell you before we bring him on, let me tell you a little bit about Dr. Paul Wischmeyer. So Dr. Wischmeyer is a critical care, perioperative, and nutrition physician who serves as a tenured professor of anesthesiology and surgery at Duke University School of Medicine. He also serves as the associate vice chair for clinical research in the Department of Anesthesiology and director of the nutrition team at Duke Hospital. He is the founder and director of the Duke Clinical Nutrition Fellowship. Dr. Wischmeyer's clinical and research focus is focused on helping patients prepare and recover from critical illness and surgery. His research interests include surgical and ICU nutrition and exercise rehabilitation therapy, parenteral nutrition, and personalized nutrition trials, perioperative optimization, post-illness muscle mass and functional recovery, and the role of probiotics and the microbiome in illness. Dr. Wischmeyer has received significant funding from the National Institutes of Health and Department of Defense. He has received numerous awards for his work from national and international societies, including the Jeffrey Silverstein Award and Memorial Lecture for Humanism in Medicine from the American Delirium Society, the John M. Kinney Award for the most significant contribution to the field of general nutrition, and the Stanley Dudrick Research Scholar Award of the American Society for Parenteral and Enteral Nutrition. In 2020, he received the Aspen George Blackburn Clinical Nutrition Mentorship Award, as well as the Excellence in Nutrition Support Education Award from Aspen. Dr. Wischmeyer has over 210 publications with over 100 citations and one paper with over 1,000 citations. Sorry, that was 50 papers with over 100 citations and one paper with over 1,000 citations in nutrition, critical care, exercise, muscle, metabolism, probiotics in the microbiome, and perioperative care, including publications in the New England Journal of Medicine. He has been an invited speaker at numerous national and international medical meetings, delivering over 1,000 invited presentations in his career. Finally, he is an advocate and lecturer for improving the patient experience and teaching providers to keep care as the focus of healthcare. Please join me in welcoming Dr. Wischmeyer to our platform today. Thank you so much for joining us. It's an absolute honor to have you.
Thanks, Lindsay. It's wonderful to be here. I'm excited to talk about one of my very favorite topics to talk about, and it's really exciting to be on this really cutting edge and I think much needed podcast, I suppose you would call it, or lecture series to reach so many people and really share good evidence and data for the things that people can do with the probiotics and the microbiome.
Absolutely. And that's exactly what we're trying to do here. And we're presenting evidence-based information for people to help create a space for that, right? Even across social media, which is a place where you don't necessarily find this. Also, I really like the fact that you're an advocate for the patient experience and teaching providers to really care, because I think that that's something that we are really lacking in healthcare. And so the fact that you share that vision, because that's something that we've also discussed on the platform, is just really inspiring. So to have someone like you here is again, it's truly an honor. I was wondering, so you're an anesthesiologist, but you also have all this interest in nutrition, the microbiome. Can you kind of bridge that gap for us? How did that kind of happen? How did you get into that area?
Yeah, so I think for me, it goes back to when I was 15 years old and I was diagnosed with inflammatory bowel disease, also colitis, and quickly went from someone who'd never thought about doing medicine in their life and had never actually been inside a hospital in their life. I think my dream was to be a soccer goalie back then, or a football goalie for those who've overseas. But with that illness and sudden hospitalization, and was in the hospital for about a year on parental nutrition for about a year. It saved my life. So I became very close to dietitians and pharmacists. They really were essential in my life and survival back then, and even now. I began to realize that I think we didn't understand GI illnesses very well. I felt like the steroids they put me on made me as sick or sicker than the illness I had, and I never improved. And ultimately, my colon perforated, and I got peritonitis and sepsis. Ended up having emergent surgery. And through my life, because of that, I've had 27 abdominal surgeries, ultimately leaving me with short gut syndrome. I have 140 centimeters of total bowel, and have had an ostomy, jejunostomy now. Some people would call them ileostomies. Mine's a little shorter, that I've had since I was 15. And so I think I began to realize, I wanted to help physicians care for patients better. I felt like I was a scary adolescent patient on an adult ward, and the physicians were afraid of me and didn't really communicate a lot of things to me that I felt they should have. And they blamed my mom for my illness, because back then they thought IBD was a stress-related disease. They always said, "Oh, what did you do wrong raising him that caused this illness?" Of course, we know that's not true now. And I most of all wanted to study things that didn't make you sicker, perhaps, than the illness you had. And so instead of studying drugs, I was really passionate about studying nutrition and the role of the parts of our body that can work with our body, sort of that improve the physiology that we were all born with, the microbiome being, of course, a critical part of our physiology, what makes us human. and nutrition as well. And so I felt like I wanted to study treatments that didn't have more side effects than the illness itself and could work with the body, as opposed to some of the toxicities of the drugs that we use, of course. And so this has been a lifelong passion. My last surgery was 2 years ago, actually. I was in the ICU for a long period of time, intubated, on TPN for 3 or 4 months with the GI fistula. And thankfully, as of about 4 days ago, have now spent 2 years without being hospitalized for the first time in my life. I've never gone longer than 6 months in my life since I was 15 without being hospitalized for a bowel obstruction or other complications. So I'm hopeful that these last set of surgeries I had a few years ago really have helped me recover. And then a lot of the things that you're going to hear me talk about and a lot of the nutrition things I talk about are things I practice every day. So nutrition and microbiome management is an everyday part of my life, both in my patient care and in my personal life. So it's something that's easy to talk about and something I care a great deal about.
Thank you so much for sharing that because, it's so important to hear someone's story and how they've arrived at a
Yeah,
certain place. And I'm sure you have just a great deal of empathy and compassion for people that have been through things like you have. And we really need more of that, I think, in the
thank
healthcare
you.
profession. Yeah, that's really beautiful. And you also have a clinical nutrition fellowship. Is that right?
We do. I think one of the major challenges I have found as I've gone further into the world of medicine and nutrition is 75% of medical schools teach no required clinical nutrition. And we have found that more than 90% of new physicians, young residents and physicians, feel no competence to discuss nutrition or any of the topics we'll talk about today with their patients because they weren't trained in it. And so I think we realized we needed to somehow begin to address this huge deficit in nutrition training in our educational system. And so we worked with a number of groups to design a nutrition fellowship, an online nutrition fellowship, using the education platforms we have here at Duke that are really, really quite brilliant for online work. And I thought to myself, if I was going to learn about a topic in the microbiome or nutrition or in these areas, who would I want to learn from anywhere in the world? So who is the best speaker, the best lecturer, the best professor on all these different topics? And I'm going to ask them to be the ones giving the lectures. And so they give each week during our fellowship, which runs every year, we have a different lecturer who then, as a lecture people watch, has an online discussion board where the fellows, and we train people from all over the world. We have people right now from 15 countries, 4 continents, many developing nations, and all different disciplines. We have pharmacists, we have RDs, we have physicians, we have all different disciplines represented. And they interact with each other over a Zoom call twice during that week period of each module with the expert or professor. So they get to know these world famous experts and they get to know each other and hear their practice. And so that's one of the most exciting parts of it. I think that's one of the things COVID has taught us to do, that we can interact online like we are today. And it's a really wonderful experience. I have to learn more from some of these people in, say, Indonesia or the Philippines or in Colombia or in different parts of the world where the resources are much more limited. And it's really neat to learn how we all practice medicine and nutrition in that setting. And so it's really wonderful. I think, we have 37 fellows this year, like I said, from all over the world. And we have just been thrilled with the response. We actually had to close enrollment. That was about the max we thought we could have and have a really good interaction each week with all the different participants, the fellows and the faculty. So we're excited to have others out there sign up and join us. We'll be starting a new fellowship again in January of next year. We have a waiting list if you'd like to get on it. We'll share some information about it and a link you can go to. And we've also started a clinical nutrition research fellowship, which will launch later this year, which is specific for those who want to do research in clinical nutrition, population health, quality improvement research within their own institutions. And we have some really great experts teaching that. And we've also started a research fellow track where we choose 2 people each year that we will mentor through a research project. And so that's one of the other things that we've started. So it's been a really wonderful experience.
Wow, that's so inspiring, and I love that it's a lot of different healthcare professionals that can join in because I
Yes,
think, your
it's critical.
conversations and just the learning, from all those different backgrounds of people, right, must just be incredible. That's really interesting, and I know as a pharmacist, I've had the comment often, "Well, what do pharmacists know about nutrition?" And it is
A
true,
lot.
it's not I mean, we can learn, but it's not covered, right, in our
Yeah,
basic education,
same
but there's
problem we have.
yeah, and I mean, we are interested in this, right? And we're interested in even trying to prevent disease before it happens, right? Which I think is
Yeah,
where where things are kind of heading, right? And there's a huge appetite for
yes.
this. We're talking about nutrition, but
Amen.
anyways, so yeah, so that's really inspiring, and I will put the link to that program in the description of this video
Great.
after we're done this session for anyone who's interested in learning
Yeah.
more. So Dr. Wischmeyer, I know that you have a special presentation for us today. So I'm
Yeah.
going to, I could talk to you forever, but I'm going to let you go ahead. And we're just really looking forward to learning from you today about the microbiome and how this affects our health.
Excellent. No, we're excited to talk about it. Yeah, and again, this is one of my very favorite topics to talk about. It's one of the most fun lectures I give. And I think it really gets at a really unique way that we need to start looking at the world. I think a lot of us in medicine spend a lot of time trying to eliminate bacteria or sh- uh, eradicate infection with antibiotics and other things that kill the bacteria that live within our body, when the reality is, and quickly I'll show my alignments of interest before I get further into it. I call these alignments because I'd like to believe that the NIH and the Department of Defense and the industry groups I work with, we're all aligned in trying to improve health for people. And so I think that's really critical. But I think rather than trying to eradicate bacteria, I think it's critical that we realize ultimately the majority of the bacteria that live in our bodies and live on our bodies are 100 trillion friends we didn't know we had. And that they're friends we want to keep around. And I think this is a very different way of thinking for most medical professionals, no matter what your specialty. And I think it really is a major part of the future of medicine, and not just preventive medicine, but also even treatment and even acute and chronic illness interventions. And I think we're beginning to see this more and more, right? I think this idea that instead of eradicating bacteria, we're going to start giving them back, whether that's through fecal transplants. There's books now that show us how we can use our own stool perhaps to improve our health. And I think the day is already here where we are actually prescribing um, bacteria and stool and poop pills and probiotics to patients to improve outcome with a great deal of evidence in many cases. And I think this is best exemplified in our now very common response to the fastest growing iatrogenic disease in the world, which is Clostridium difficile colitis. I just saw another patient with it today. When I was a resident, I would see one patient with this a year. Now I see multiple patients with this a week. And I think that is due to the prevalence of these broad spectrum antibiotics that are so effective at killing everything in our body, not just the offending organisms we're trying to kill. And I think this is one of the first and best stories that really exemplifies the life-saving effect of giving back bacterial interventions. And this was a really unique story. It was all over the news some years back. It was a story about a young woman named Caitlin. She was a 20-year-old who was otherwise completely healthy and got in a car accident. And she was improving from her car accident, but unfortunately, because of the antibiotics she needed to get during her hospital stay, she developed very severe resistant Clostridium difficile and went through multiple rounds of antibiotics without recovering, and was getting to where it was likely to take her life. And as a last ditch effort, the physicians gave a stool transplant from her from her mother. And ultimately, it immediately eradicated the disease and truly saved her life. And so it was really, I think, one of the true success stories that now we have many like this. And this is a common intervention for a range of illnesses, but in particular, Clostridium difficile when it gets severe. And I think it brings us to the point that perhaps due to the whole range of things that we do that are negative to our gut health and to our bacterial health, whether that be antibiotics, whether that be illness or surgery or cancer, infectious disease, COVID, all cause sort of a complete loss of the normal bacteria that live in our guts. which is really critical for who we are as humans, as I'll show you. And the idea that I think we need to be resodding the lawn, and giving back the normal sorts of things that make us human with probiotics, stool transplants. The kids at MIT will sell you their poop pills, and they'll tell you that it'll make you smarter, although the FDA has some qualms with that. But even sort of these targeted stool pills that have targeted probiotic species in them, I think have a really great future when done properly. And why would this be so effective for us as humans? It's hard for a lot of people to grasp, but the reality is it's important to remember that the world we live in is largely a bacterial world, a microbial world. Most of the organisms that live on the earth, more than 99% of them are bacterial. We as multicellular organisms from humans are a very narrow part of the sort of living world of the earth and our bodies itself. And until recently, most of these microbes we didn't know anything about. We couldn't grow them on a culture, but we've got new techniques now that have allowed us to discover them. And why should you care about your microbes so much? Well, the reality is they affect every part of your life. And have you ever wondered why, when you go into the woods or go camping, you get bitten by all the mosquitoes and your wife, husband, friend doesn't get bitten by the mosquitoes? It's not because you're sweeter. It's because there's a microbiome that lives on your skin that either attracts or repels mosquitoes. So again, even the more basic things we do every day are largely affected by our microbiome, so much so that in fact, our microbiome may ultimately determine who we have sex with and or marry. We actually know this is definitely true if you're a fruit fly. So fruit flies' mating behaviors and preferences are based on their gut microbiome makeup. And so someday we might actually find out this is true for humans too, and that would be shocking, but it's definitely true in some of the other studies of species that have been done. And so human studies like this are going on in humans as well. And how do our bacteria do all that? And I think this is a critical question. And the question is, what is it that makes us human? And some people say, well, our cells make us human. Well, there's 30 trillion human cells that live in our body at any given time, and 38 trillion microbial cells in all of your intestines and in your body right now. So you're actually more than 50% bacterial from a cell makeup point of view. So then many people say, well, maybe it's our genes that make us human and who we are. Well, we all have about 20,000 human genes in our body at any given time, but we have as many as 20 million microbial genes. So we are actually about 1% humans genetically and 99% microbial in terms of the genes that make up who we are as humans. And so finally, we're able to answer and begin to answer, and we have begun to answer many questions about who we are as microbial cells. And that's because as opposed to the past where we could only culture a very small percentage of the bacteria that lived in and amongst ourselves as humans, now with ribosomal microbiome techniques, ribosomal RNA sequencing, we can see virtually all of the bacterial species that live in our body. And so the NIH and the US government spent about $250 million sequencing in the big microbiome project, a few hundred medical students, and it generates a ton of data like this. It's actually about $173 million spent just generating terabytes of data about the microbiome of a normal human. And so what we do is we take data that looks like this and we can map it. And I think it's key to realize that in all of our bodies that different parts of our bodies have very different looking microbiomes. So the mouth versus the gut versus the vaginal canal versus your skin all have very different microbiomes. And these are all really critical to many parts of what make us human and how we develop as human beings. And so when we take all those A, T, C's, and G's, we generate plots like this. And this is data from the NIH Microbiome Project. And so under the microscope, virtually all these bacteria look very similar. But when you plot each individual person as a dot on this map, as you think of it as a map, like continents are on a map, you can show the differences in different parts of the body. The green dots are the mouth, the blue are the skin, the purple are the vaginal canal, and then the fecal microbiome constituents, the different bacteria that make it up, place them at different places on a map, much like continents on a map. And in fact, there's dramatic differences, as you might be able to see on these maps, from the mouth and the gut. In fact, there is a bigger difference in the species that make up the bacteria in your mouth versus your gut than there is all the bacteria that make up the coral reef, say in Australia, versus the plains of the US. So there's a bigger difference in bacteria in the 3 feet between your mouth and your gut than there is in thousands of miles in the different ecosystems on Earth. So we as humans are very diverse, and this diversity is very important. And is this stable? Does it change over time? Well, one of the people I work closely with, Rob Knight, he has sampled himself and his kids every day with a swab, and then he analyzes it. And so actually, this is a neat map that shows how the fecal microbiome changes in the skin and the mouth. All these different microbiomes change day to day over months to years at a time. And so you can see that the different bacterial makeup of the different parts of our body stays very stable over time in a healthy individual. We will see in a bit that that changes dramatically when we become ill, and it creates risk for us of infection that we can do something about. So where does our microbiome come from? Well, if you have a dog at home, as Lindsay was talking about before, you have a different looking microbiome than if you don't have a dog at home. But the reality is our microbiome comes to us from our mother at birth. And for many, many, many years, of course, we were all born by vaginal delivery. So in fact, our entire microbiome, including our fecal microbiome, resembled mom's vaginal secretions. And it wasn't until recently that we began to be born by C-section. The unfortunate or challenging part of being born by C-section is when a baby's born by C-section, all of the microbiome of their gut and their mouth and the other parts of their body resembles mom's skin. And so we know that children born by C-section have more asthma, more allergy, more obesity, more ear infections, for instance. And so one of those reasons is the immune system doesn't develop the same if you're born by C-section versus vaginal delivery. And so one of the microbiome researchers that I work with, actually, when his child was born, kicked everybody out of the delivery room, and when the child was born by C-section, and smeared mom's vaginal secretions all over the baby to be sure their children would develop with the normal vaginal flora. So it's probably the only thing you remember that I've said today. But if you have a baby by C-section, I would encourage all of you to take some of mom's vaginal secretions, put it all over the baby. So anyway, it's one of the things that clearly defines how our immune system develops and potential risk for illness and things like obesity later in life. So you've seen a child develop psychologically, you've seen a child develop physically. What about how a child develops microbially? And so again, this is one of those wonderful experiments where one of the microbiome researchers sampled their child every day from birth to the first 900 days of life. And so this is a child's fecal sample in yellow, and you can see it resembles mom's vaginal secretions quite remarkably. But ultimately, right, we would like, as the child develops this, to begin to resemble a normal fecal microbiome. And so if you watch over time, this is how a child develops over time microbially. And you can see it takes many, many, many days for the child's fecal sample to begin to move towards a fecal appearance. It looks like mom's vaginal secretions for much of the time. And then slowly but surely, you can see it's beginning to develop and move towards a normal fecal microbiome. Now, this child got an ear infection when they were about 2.5 and got antibiotics just for a few days. And you could see what happened. This is a few days of antibiotics. It caused a severe regression in the microbiome back almost to what it looked like at birth. And thankfully, this child recovered, but not everyone who gets antibiotics recovers a normal microbiome. And so this is the risk we take every time we take antibiotics. And so again, you can see that again, a dramatic change, even from just a few days of antibiotics in what makes us microbially normal. And then ultimately, this child did recover, and ultimately their microbiome then began to correct to normal. And that's the question, do changes in the microbiome matter? Like, we know that antibiotics in the first 6 months of life increases our risk of obesity, of allergy, of heart disease, and other infectious diseases. So clearly the preponderance of antibiotic use in children is a challenge. The other interesting thing, of course, is looking at the stool of a patient, we can tell with 90% accuracy whether that person's obese or not obese. Now, you might not think that's very useful because you can just look at someone and assess if they're obese. But if you looked at all their genes, you'd only be able to tell with 60% accuracy. So as it turns out, we believe the microbiome is more important for obesity than your genetics is. And I think this is best exemplified by some of these really unique experiments that are done that shows how you can change a person or a mouse dramatically just by taking the microbiome from one individual and give it to another. So there's lots of experiments that are done with germ-free mice. These are mice that are born and then never exposed to any bacteria. So they're completely sterile of bacteria. And if you take these germ-free mice and you take the stool from an obese mouse and give these bacteria to a germ-free mouse, those mice become obese. And not only do they change their metabolism, but they actually eat more. It changes their brain, it changes how they think, and that's a different topic for a different day, but it affects the brain a great deal. It affects all kinds of different parts of metabolism and how much they eat. Whereas if you take the stool from a thin mouse and you put it into the germ-free mice, the mice remain thin. Now, the next question is, can we recreate human disease in a germ-free mouse with a human stool? So if you take the stool from an obese human and give it to a germ-free mouse, the mouse also becomes obese. And so again, the idea that we can take the stool, and you were talking about twin to twin differences in the microbiome, there's experiments going on right now at Washington and other institutions where they are taking the stool from a thin twin pair and giving it to the obese twin to try to improve obesity. And so there is promise that we can change the characteristics of a human by taking the stool from one human, giving it to another. And this is true not only for obesity, but malnutrition. If you take twins, one who's healthy and one who's malnourished, they did a lot of this work in Africa and in some of the refugee camps. If you take the stool from a kwashiorkor or malnourished child and give it to a germ-free mouse, the mouse becomes malnourished. And so our microbiome is critical to generating amino acids and to generating nutrients. And when it's deranged, it creates malnutrition as much as it can create obesity. And so I think this is really compelling that the microbiome makes up who we are. And I think one of the things that's really critical is we as humans in the US and in industrialized nations have dramatically changed our microbiomes just because of our diet and our exposure to antibiotics. If you look at the microbiomes of tribes in different parts of the world that have not been exposed to Western medicine, not been exposed to a Western diet, they have a much more diverse and varied microbiome, a much healthier microbiome than those of us who lived in the Western world. And although getting to the infection part is the focus of this lecture, we'll get to in a moment, as we've eradicated a lot of or reduced the incidence of infectious diseases, we have at the same time caused an exponential rise in autoimmune disease. So again, our body needs to tolerize to bacteria through our lives, and it's critical we maintain a normal microbiome. And when we don't, by eradicating it with bacteria, we create autoimmunity. In fact, the illness that plagued me when I was a child is an inflammatory bowel disease we think that is related to changes in the microbiome that go on at a very young age, potentially. And so I think this is a key thing we need to think about, that yes, antibiotics have their use, but I think they also have created a whole new world of autoimmunity and other diseases. So can we change the microbiome to cure disease, things like infections and respiratory diseases? And what's in our patients' guts is the key question. Can it help them or can it hurt them? And I think it's critical to realize, right, that when we have infection or chronic illness, cancer, surgery, ICU, COVID, it creates imbalance. And it's critical that we restore that balance and we resod that lawn. And we give back things like probiotics, which I think are the easiest way to do it, especially for those of you at home. But even in the hospital, we do the same thing. And I think probiotics really have exploded recently, right? They're in lots of our foods. There's lots of ways to get them. There's books about them. You can learn all about them one way or another. I'm not sure this is the best way, but it's one way. But I think if we're going to treat our patients and treat ourselves with probiotics, we need to be very thoughtful about it, much like if we were taking any other medicine or intervention. And so if we're going to make a choice to use a probiotic, it needs to make mechanistic sense. And so what's the science for taking a probiotic every day or taking a probiotic to help recover from illness? Well, the science actually is quite robust. And this is a neat figure I'll talk about that was generated by John Alverdi, who trained in the same lab I trained in at University of Chicago, in the lab of Eugene Chang. He's a world-renowned gastroenterologist and researcher in the microbiome and GI world. And one of the things that John Alverdi discovered, who was a surgeon, actually trained me, was that bacteria actually sense our stress and talk to each other. And the presence of stress, a poor diet, exposure to antibiotics, exposure to a poor diet, but especially illness, stress, and antibiotic stress causes the normal bacteria to go away. It's a little bit like when a hurricane hits New Orleans, for instance, all the regular people that live there evacuate and they leave. And so what happens then is looters come in. So sort of bad folks come in and start looting the shops in the town, and they bring their bad acting friends, and they start looting. And so what happens when our gut loses its normal flora, whether it be from antibiotics or other illnesses, bad bacteria like Pseudomonas that live in all of our intestines right now. There's Pseudomonas in all of your intestines right now, but they sit quietly and they wait for you to be stressed or for all the normal bacteria to go away. And then they come out and they attack us and they express toxins and they bring all their bad looter friends in with them, their other bad bacterial friends, and they cause invasion and illness. And so if we give back the normal bacteria, i.e. we bring the normal people that live in New Orleans back to the city and to their houses and to the town, the looters go away. And it's the same for bad bacteria. If you use probiotics to bring normal flora back, they suppress the bad bacteria and they turn off the toxic and aggressive behavior of things like Pseudomonas and other infectious disease. So this is why you want to give commensal flora, perhaps take this every day, just to keep the bad actors at bay. And so prebiotics play a role in this as well. Prebiotics are the food that normal probiotics eat, and they have a pretty significant effect on our immune system and how our body responds to infection, how our body prevents infection. And so again, having a good fiber in most cases is really critical as well, because when that's not there, the bad bacteria can act up and they can erode through your gut barrier and they can cause disease and autoimmunity and lots of other complications. Your gut has a huge immune system that needs to be maintained, and that is critical that it's done with a probiotic and then also good prebiotic fiber as well. So science is a start, but it's not sufficient. So the next question we often get when we give probiotics, say, to treat an infection or to prevent an infection is it going to cause harm? Is it going to cause infections, right? People say, oh my gosh, I don't know if I want to take a probiotic or a bacteria because I might get an infection. So the AHRQ, the US Health Quality Research and Quality Group, did a huge study a number of years ago, 645-page study, looking at every probiotic study ever done to that point. And they were sure they were going to find risk and harm and infections and complications from it. They studied 622 probiotic studies, and they found there was no risk of adverse events or infection in healthy people or sick people from probiotics in these larger studies. So you can see that the confidence interval there falls right on 1. So it doesn't have any risk at all. And in fact, if you looked at the people who were sick when they took a probiotic, there was a trend towards less adverse events and less infections in high risk or critically ill patients. So there was actually a trend to benefit if you were sick and you took a probiotic. So this was not what the study thought they would find. And they really say they were surprised and I think encouraged to find that probiotics were quite safe and in fact look to possibly be beneficial, especially when you're sick. There's one study that people often bring up. I've actually had pharmacists bring it up, called the Propatia trial, that did show risk. It's one of the only studies that's ever shown risk from a probiotic. It was a study of pancreatitis. And for any of you out there who care for pancreatitis patients, you know that they have really severe ileuses and their gut moves very slowly. And this study did probiotic therapy a little bit different. They took sort of a 9-component probiotic that hadn't been studied too much and a fiber, and they gave it to patients with pancreatitis in a small bowel feeding tube. So it escaped the stomach. And unfortunately, there were 2 groups, and when they did their interim analysis, they noticed there was more mortality in one group than the other, and they should have stopped the study, but they didn't. They thought the probiotic group was helping, and they said, we'll have a better study if we continue the study. Well, the reality was the group getting the probiotics in the post-pyloric tube with the fiber actually were dying more. They were developing, I talked to some of the surgeons that operated on the patients in this study, and they would have prebiotic fiber balls just outside the feeding tube that hadn't moved, where bacteria could thrive and erode through the gut wall and causing perforation. And so again, there was lots of ethical and design concerns that became quite famous in the medical ethics, research ethics literature. But the reality was giving a combination of a fiber and probiotic to the small bowel in a ileus causing pancreatitis led to local bowel ischemia. And I think one of the real lessons here is you should never give probiotics or prebiotics for that matter post-pylorically. If you give them to the stomach and the stomach doesn't move forward, you get reflux, you'll get gastroresiduals, and they won't sit in the bowel for days and days. The stomach is good at A, acid will kill the bacteria if it sits there too long, and B, it won't move forward and they won't tolerate it and they'll get stopped. So you should never give probiotics post-pylorically. I think that's what this study taught us. And so this was the only study that showed a risk, and I think we can avoid that risk quite easily in sick patients who we don't give them to post-pylorically. But in the other 622 trials, there was not an increased risk from probiotics, and they were very, very safe. So what's the clinical data for the benefit of probiotics? And so there's a whole wealth of data we can draw on here, but I think this is one of the biggest and largest meta-analyses. So when we give antibiotics for infection, one of the common side effects, many of you have probably experienced it, is we get diarrhea because we eradicate the normal bacteria. And in this 12,000 patient, multiple RCT meta-analysis published in JAMA, it was shown there was about a 40% reduction in antibiotic-associated diarrhea when a probiotic was given. And it really didn't seem to matter which probiotic you got. As it turns out, right as I told you, commensal bacteria, normal bacteria, will outgrow a pathogen pretty much every time. And so as long as you gave something that was a commensal normal bacteria, a healthy bacteria, it would eradicate the bad bacteria causing the diarrhea. And that's definitely true for C. diff. C. diff's a weak little organism. But when all the other organisms get run out, much like the hurricane, C. diff can thrive. And that's why this is so effective against antibiotic-associated diarrhea. And then one of the other really exciting trials that was done shortly thereafter was an NIH-funded study looking at giving lactobacillus GG to patients at risk for ventilator-associated pneumonia. And they did two interesting things in the study. One, they smeared a capsule in the mouth because the oral pharynx is probably the source of many of the pneumonias our patients get in the ICU. And they gave one capsule to the stomach. And they found a 50% reduction in ventilator-associated pneumonia in the intention to treat group, so the most rigorous group, and it reduced C. diff. So this was a really compelling trial that I think showed a great deal of promise. And ultimately, this led to my group and others doing meta-analyses that showed that multiple different probiotics could reduce infections and ventilator-associated pneumonia in ICU patients. This was a large randomized controlled trial meta-analysis that's in a lot of trials. And the sicker you were, the more likely you were to benefit, which makes sense. The sicker you are, the more deranged your microbiome is. And there were a lot of studies that contributed to this. This ultimately led then to a large randomized trial that was done, actually with a Canadian PI. It was funded by the CHR. And they looked at 44 ICUs across the world. And in this case, they only gave the probiotic to the stomach with the hopes of reducing ventilator-associated pneumonia. They thought it would be simpler. And they had, again, the lactobacillus GG versus a placebo. Unfortunately, there were no differences in ventilator-associated pneumonia in this study. And so I think it hints that probably using some of the probiotic in the mouth is probably very important. But in general, this one did not show a benefit on outcomes. So I think we need to be more thoughtful, perhaps give a probiotic with a prebiotic, as we'll talk about in a moment, and some other very successful studies. There were opportunities, there were a very small number of patients where the LGG that was given showed up at a non-sterile site, whether it be a blood culture or somewhere else, as a sole organism, potentially as an infectious organism. LGG is typically not a particularly bad pathogen, but it did seem to be able to, in the rare patient, escape and show up in the blood or other places. So a large meta-analysis was done using this data and combining 65 trials now, and there still was a positive signal on reducing ventilator-associated pneumonia. But I think if you're going to do this, it's key that you give the probiotic not just to the stomach, but also to the mouth. You smear a capsule in the mouth, which is how we encourage our nurses to do it at Duke when we use these. And this showed benefits on a range of different things: ventilator-associated pneumonia, healthcare-associated pneumonias, potentially mortality and ICU length of stay. And duration of mechanical ventilation. So there were trends to benefits in all these, but I think we continue to refine our data and how we give our probiotics to show the best ways to do this. So I think just giving it to the stomach probably is not the best avenue. I think you need to treat the stomach and the mouth. There are studies in elective surgery and in trauma that reduce infection and treat sepsis as well, and this data continues to grow. Our guidelines basically say, our nutrition guidelines and our critical care guidelines say these should be safe and should be used thoughtfully in our patients, but we need more data. And then, of course, use of dietary fiber is also very important to the recovery of our microbiome over time. These are patients. It was an interesting study done that compared omnivores and vegans to exclusively enteral nutrition patients that were not getting any fiber at all. And as you can see, proteobacteria, which are the pathogens, were able to sustain themselves and continue to exist in the gut in the patients that weren't getting fiber. So there may be a key role of a prebiotic fiber given with the probiotic in patients to optimize the benefits of a probiotic, especially after antibiotics or when a patient's sick. So what about probiotics and C. diff? I mean, this would be an area where it naturally should work to prescribe a probiotic in a high-risk patient. There's large Cochrane analysis looking at C. diff-related infections that have shown significant benefits on reducing C. diff diarrhea, which is the actual disease symptom. There's a 60% reduction in risk of C. diff when a probiotic is used. And this is true especially in high-risk patients. So patients that have had multiple antibiotics or broad-spectrum antibiotics, people that are at very high risk of C. diff, this benefit gets even greater, especially in these trials that looked at the highest risk patients. So there's really a potent benefit of a probiotic in patients that are high risk. And this was true in adults and kids, in patients and outpatients. Different species worked. And again, different qualities of trials showed the same result. So again, the reality is C. diff is a weak little organism that when outcompeted by any commensal bacteria, healthy bacteria, is going to lose. And that's why this is so beneficial, and already clearly not only probiotics, but fecal transplants have shown that to be true as well. In fact, there's more than 90% C. diff cure rate with the use of stool transplants. And there's some really cool microbiome data that's just come out not so long ago, looking at how effective C. diff treatment is with a microbial transplant or a fecal transplant. So again, this is that normal microbiome appearance from the healthy subjects. These stars are the fecal samples from a group of patients at the University of Minnesota who had C. diff. So you can see their fecal samples stop resembling fecal output and start resembling other parts of the body, start resorting like to the skin and to the mouth. And so this is how abnormal the gut looks after it becomes ill or is treated with antibiotics. So a number of these patients, the stars, received a fecal transplant. And the donor is shown, their microbiome makeup is shown at that bottom star. And you can see in this next little chart what happened when the patients in the stars got the fecal transplant. You can see their microbiome immediately returned to a normal fecal appearance. And these lines represent the change over many days after it. So not only does it prevent the derangement of the microbiome and cure the C. diff, but the microbiome of the patient goes back to normal and stays normal. So this is a long-lasting effect and benefit it has to the patient. And so that's why I think this is so critical. In fact, these studies and others are beginning to encourage us to say to people, you should all be putting your stool while you're healthy in your freezer and keeping it. There's a large study going on in cancer where they're taking stool from patients before they undergo bone marrow transplant, they're freezing it, and then giving them a fecal transplant after their bone marrow transplant to restore their microbiome. And so maybe all of us should be storing our stool now. But I think the most important trial in terms of treating infection and the landmark trial of any probiotic study ever done is one that was published recently in Nature. So those of you who may know, Nature is one of the highest impact, most well-respected, most renowned journals in the world. They only take the very, very, very best of all studies done. And this was a study done in healthy newborn infants in India. And it was done by Pinaki Panagrathi, who's now at Georgetown. He was at Nebraska before this. The study got millions of dollars in NIH funding to be done. And they looked at giving lactobacillus plus a prebiotic, FOS, fructooligosaccharide, in healthy newborns versus a placebo. And they looked at them over multiple days, and they looked at viral infections, bacterial infections, sepsis, and death. And that was the primary endpoint. And in this large study of healthy children, getting a pre and probiotic significantly reduced sepsis, respiratory infections, and death in the healthy children that got them. This was a hugely compelling study that really shows that you can massively reduce the risk of infection in healthy people using a simple probiotic-prebiotic combination that is easily available to anyone really in the world. It's an inexpensive intervention. And in fact, every 27 people who got one of these probiotic interventions, prebiotic interventions, a life was saved. So again, significant reduction in the combination of sepsis and death in the treatment arm, significant reduction in both bacterial and viral infections and lower respiratory tract infections. So again, compelling data. And it began to make us think that not only could this help in this setting, but perhaps it could help in other viral infections like COVID-19. And that ultimately led us to begin to do some research, which we'll talk about later, because I think the reality is all of us were a setup for the COVID-19 pandemic and for many of the different viral pandemics like influenza and others that are going on, because many of us live in the industrial society we live in, where we eat sort of a very poor Western diet. We take antibiotics from time to time. We have other illnesses. All these illnesses significantly affect your microbiome in a negative way. They cause dysbiosis, and this sets us up for immune dysfunction and infectious risk. And then COVID-19 appears. All of us are set up to be susceptible to COVID-19, and that's why people with these other comorbidities got sicker. And if you hadn't received a probiotic, or if you didn't correct your dysbiosis, we found that you had much more inflammation in your lung and other organs. Your ACE2 expression, which is how COVID infects you, went up. And in fact, it appeared that patients with severe dysbiosis had much worse outcomes from COVID. And so we hypothesized that not just in COVID, probably other viral illnesses, that if we could correct that with a probiotic, we would reduce the severity of illness, we would reduce the infectious rate, and we would improve outcomes in these patients. And so, is there other literature for probiotics improving viral respiratory infections? And the reality is there is. This is a large, again, Cochrane, the most well-respected review of the data that exists, 13 trials, 3,000 patients. Probiotics reduce an upper respiratory tract infection episode in healthy individuals by 47%. They cut your risk in half. So if all of you took a probiotic every day, you would have your risk of getting a viral infection, influenza, and all the other things that people get throughout the year that lead to a lot of the complications and illnesses that we see. And in fact, it reduced the duration, not just people getting sick, but when you did get sick, the length of time you were sick went down by about 2 days. So this is just one large meta-analysis. There's another large meta-analysis, now 10,000 patients, that shows that symbiotics, pre and probiotics, like given in that Nature paper, reduce respiratory tract infections about 16% in all comers, kids and adults, and about 32% in adults alone. So you can significantly reduce your risk of getting a viral respiratory infection if you take a probiotic daily. And so again, this data was quite potent. Based on that Cochrane analysis, an economic modeling group said how much money could the US save if everybody took a probiotic every day? And what they found was the cost savings for healthcare payers, insurance companies and other groups, would be about $370 million. This is like Medicare and other groups. It would avert 54 million respiratory tract infection related sick days, 54 million. It would reduce antibiotics use by about 2 million antibiotic courses per year. It would reduce work absences by $4 million and save the US about $1.4 billion for what would really be a very minor investment. So again, the potential ramifications of us improving our microbiome with a simple probiotic are pretty immense. And so this was really compelling data, I think, from a healthcare systems point of view and preventive medicine point of view. So what we felt like when COVID started, our research group said, gosh, there's all this data that shows how well a probiotic can prevent infection in the respiratory tract. We should study that. So we initiated a study called the PROTECT-DHC trial that looked at probiotics to eliminate COVID transmission in people who had someone in their house who tested positive for COVID-19. So we called every patient at Duke over about a 1.5-year period who tested positive for COVID at Duke and said, we would like to treat everyone that lives with you, whether they're 1 year old or 99 years old, with either a probiotic or a placebo to try to prevent other people in the home from getting COVID when they're at high risk because someone in your home has COVID. And so this was the trial that we did. It was called the PROTECT-DHC trial. And we basically sent out probiotics overnight. We overnighted them to people. This was all done remotely. And then we had them weekly do a log of their symptoms. We had them do a nasal swab and a stool swab that they sent back to us. And then we followed them for out to 60 days to see if they got COVID and what their symptoms were, if anybody else in the house got infected. And so again, we used a lactobacillus GG, which is the same lactobacillus that was used in the large ICU trials. And then we had a control as well. We got an FDA IND for this. That's one of the challenges in probiotic research. You have to get FDA INDs now. They treat them like a drug, which makes the research harder to do, but this actually was very easy to get. Those of you who are considering doing probiotic research, we got this in a few weeks. We collected a lot of data over time, including these swabs. We enrolled not only at Duke, but we used Facebook ads. We used social media to recruit. We did a bunch of TV news interviews and some other strategies that we used. And we really wanted to look as, what is the baseline microbiome's effect on risk of COVID-19? Can probiotics prevent it? Can it treat it? Does it change the risk of getting it? And so we just published our paper actually just a few months ago. We ultimately enrolled 182 participants, randomized for those 28 days. And again, our primary outcome was development of symptoms and or development of COVID-19. And we wanted to look at the microbiome as well. And what we found was, again, we had a number of different groups that we stratified people into. One was, if you got into the study at all, if you signed the consent, we counted you as someone who was enrolled. And that was our true intention to treat group. If you actually took the probiotic or the placebo, you then moved down into a modified intention to treat group. And then the last group was, were you asymptomatic? Did you have any COVID symptoms at all when you started the study product? versus those that already perhaps had symptoms. Again, it took us about 36 hours to get you the drug, and some people developed symptoms in that interim. And what we found was across all of these groups, using a probiotic reduced symptoms of COVID significantly, whether these were people just in the intention to treat group or if these were people that were asymptomatic at the start. And the data actually got stronger in the more rigorous groups. The p-values were stronger in the groups even that were the intention to treat setting. And so again, this significantly reduced COVID symptoms by about 40 to 50% in the patients. And in fact, the reported COVID diagnosis, the raw COVID diagnosis nearly was significantly reduced as well. And what was reduced was symptoms as you see over time. And so you can see over time, if you follow them out to 28 days, there was a major reduction in the symptoms people suffered with their COVID. And the time to COVID diagnosis was significantly improved. So again, far less people over time developed COVID-19 if they took a probiotic, and this was statistically significant, as you can see in the groups, with the red being the placebo and the blue being the active. And again, this is a graph showing people who did not get COVID. So very few people who took a probiotic got COVID, and this was significant over time. And so again, then we also wanted to look at the fecal microbiome to see if we could see the LGG showing up. And this is one of the ways we tested if people were taking the probiotic, right? Again, that was hard to control for. So we actually measured LGG in the fecal microbiome, and you can see it was significantly greater in the probiotic group than it was in the placebo group. And so again, I think people were pretty good about taking it in most cases. And in fact, we're still working on this data, but there were significant changes in the fecal microbiome, both in patients who had COVID versus those that didn't, and in patients who got the probiotic versus those that didn't. And these changes actually were quite dramatic, quite statistically significant. So again, in our intention to treat analysis, patients who got a probiotic were much less likely to develop symptoms. And over time, they were much less likely to diagnose themselves positively for COVID. And so we felt like, well, really, this was an initial pilot trial of this, was very promising. And I think this really encouraged us that this could be a preventative measure for COVID and perhaps other viral infections, much like the meta-analysis data have shown. And again, we felt like patients were taking it. We could see the LGG in the fecal microbiome samples, and there was significant relationships of the microbiome to your risk of COVID and the effect the probiotic had on your microbiome itself. So again, probiotics seem to have a major impact on reducing the risk of viral, bacterial, and other respiratory infections like COVID-19, but others as well. And this, I think, is one of the motivations we have to encouraging people in their day-to-day lives to take a probiotic as part of their regular routine, much like you might take a multivitamin or anything else that you would take to stay healthy during your regular life. Finally, to close, what about in ICU patients and our sickest patients? What happens to the microbiome, and can we equate that with outcomes? And so, again, we embarked upon a project at Rob Knight to look at our very sickest of patients, because no one had ever actually done this when we started. And so this was the first publication of what happens over time to the microbiome in our very sickest patients. It's called the ICU Microbiome Project. And it looked at 4 centers in the US and Canada. We teamed up with a group in Canada to do this. And basically what we found was all the patients to get into the study had to be on a ventilator for 48 hours. And you can't be on a ventilator in North America for more than 48 hours and not get antibiotics. Every patient got them. So this is universal. And you can see, if you look at the American Gut are volunteers that Rob Knight got to send them their stool, and people would pay Rob to send them their stool, and he'd send back their microbiome makeup. He made millions of dollars doing this. The US government spent $200 million sequencing 200 people. Rob Knight's made $5 million sequencing thousands of people. It's really brilliant crowdsourced funding science, but those are the normal folks in the American Gut in red. And then the ICU patients you can see are dramatically different in their microbiome makeup in blue. The other thing that we found was, so firmicutes are one of the most common healthy organisms that live in our guts. And you can see the redder the dots, the more of these healthy bacteria you have. You can see in the ICU patients, the big dots, they're almost all white. That means the firmicutes are gone. So there's a dramatic loss of normal bacteria, much like the regular folks that live in New Orleans, they all go away. And that's when the looters come in. And so we saw a dramatic loss of normal bacteria in the ICU subjects. The other thing we saw, much like in that C.diff study you could see the normal microbiome layout in the small dots there. And if you look at the ICU patients in the big dots, you can see the mouth, the gut, and the skin. begin to look the same. You can see there's like a coming together, a loss of diversity, and a loss of differentiation from different parts of the body that should normally exist. So your stool and your mouth begin to look the same, which is a little disturbing. The other thing that we saw was, you could see in the top two graphs there, those red lines represent, each of those lines represent a patient. They had a much greater abundance of pathogens, the proteobacteria, which are the red, versus the healthy subjects, which are on the bottom graph there, that have very little red and largely are made up of the blue and the green, which are firmicutes and bacteroides. And that's what we should look like. And that's lost dramatically at both very early in ICU and at the discharge from the ICU as well, the pathogens overgrowing. We found significant normal bacterial loss. Fecalibacterium presnutzii is a key target in IBD. It makes short-chain fatty acids, which are very healthy for us. Massive losses of this, and this is a major target for treatment to give back. It's an anaerobe, it's hard to grow, but it's still something we look for as a treatment intervention. And then we had a great enrichment in the stool of Staphylococcus, Staph aureus, Proteus, and other pathogens. And so these began to show up in the stool in large amounts. In the mouth, the same. We lost normal bacteria, and staph and mycoplasma that cause pneumonia were greatly increased in patients' mouths. And you can see there's this great increase in the stool of many different pathogens that cause infections when we get sick. And we lose many of the healthy bacteria, especially Fecalibacterium. We also lose diversity. So that those black lines are what a normal person's diversity looks like, the diversity becoming less. And the red is quite dramatic. In fact, if we were a hunter-gatherer species, our lines would be almost straight up and down. So a Western diet is not totally normal either. And I think one of the most compelling things we saw was that ICU admission, the most common organism in the gut, made up maybe 25% of all the bacteria. But by day 2 or 3, 95% of all the bacteria in the intestine could be one organism. It was often Acinetobacter or Enterococcus. And again, so pathogens completely take over the city, much like the looters take over the city in New Orleans when a hurricane comes. And they can be 95% of all the bacteria in the gut very quickly. There's a crash in diversity. So some quick results. There were some associations of Klebsiella and other skin bacteria actually in patients who died. There were some associations with bacteremia and other infections with oral mycoplasma. That's why I think smearing probiotics in the mouth when ICU patients is very important. That's where a lot of these pathogens are living. And even ARDS had some correlations. One of the questions we got was, is this just an effect of antibiotics because everybody in the ICU gets them? Or is it actually affected the illness itself? And what we found was we could generate an antibiotic pressure score based on the antibiotics the patients got. And what we found was, using a published antibiotic pressure score that was taken from a Lancet publication, we found that in the mouth, the changes in the microbiome did have a significant relationship to the amount of antibiotics and the broad spectrum nature of the antibiotics that a patient got. But in the gut, that was not true. So the changes in the fecal microbiome don't seem to be related to the antibiotic pressure, but seem to be related to the illness itself. So just becoming ill causes this loss of gut health and the fecal microbiome. So again, this seems to be more than just antibiotic use. And the more diverse your microbiome stayed, the shorter your length of stay was. So again, coming in with good diversity in your gut and maintaining that seemed to be really critical to getting out of the ICU sooner. One of the key points of this study was to develop targets. We went to the NIH with some large trial ideas, and they said, we don't believe that one probiotic is going to treat everybody. Come back to us with targets that may be beneficial in an ICU patient. And that's where a lot of these depletions of these healthy bacteria, like Fecalibacterium, became targets for us for future probiotic studies. And these are very similar targets that are seen in IBD and Crohn's disease. So it's interesting, these diseases may be very similar. So again, the magnitude of change in diversity seemed to correlate with mortality, and we're continuing to do research in this area. So as I finish, what have we learned so far? I think the key is a healthy gut is a diverse gut, and that's true for all of us, well, sick, healthy, or not. And that illness in the Western diet and antibiotics leads to significant loss of diversity that's associated with illness, infection, and poor outcomes. And we need to correct this every day in our lives and in our patients probably as well. And I think if we can do that, we can change the world. We can change the microbial world that our bodies and those of us that live in the world live in. And so I think the idea that we can use poop and prescribe bacteria rather than eradicating bacteria has great promise, and already is showing a significant amount of data for benefit in reducing infectious complications like viruses and other infections. And in fact, we should be resodding the lawn that much of what our Western diet and antibiotics and illness do to us, and we can restore balance. And now with the new microbiome techniques becoming so infinitely inexpensive, we can make it so simple a child can do it. And so as I finish, I want to thank Rob Knight and his group that helped us do a lot of this data. I will make a last plug for our Duke Online Clinical Nutrition Fellowship. We would love to have the chance to see more and more of you in our fellowship and get to know you and work with you and improve nutrition education nationwide. And like I said, we actually have an online clinical nutrition training program as well for those of you who are academic folks. And we're actually even training fellows specifically with mentoring and other tasks. So with that, this is what we're left to do. I encourage all of you to follow on Instagram, X or Twitter, LinkedIn. I post lots of new information, lots of the new research data that both we do. We have a big paper coming out on the role of fiber in illness, whether we should be giving it or not to our patients, and a lot of new data coming out on the probiotic and the microbiome front. So you can see all of that on Instagram and Twitter. And then I'm happy to send these slides or any of the papers that you've heard me talk about. If you want to email me at this email, I'm happy to share these slides or any of that information with any of you. So thank you again. It's been wonderful talking with you, and I look forward to your questions.
Wow. Thank you so much, Dr. Wischmeyer. That was just an incredible amount of information. But truly, I love the stories as well. And the analogy to New Orleans, that was very helpful in digesting all of that. So Graham, how are you doing, Graham?
Oh my gosh, that was like I just found myself just staring at the screen the whole time. Like that was amazing. Those
Yeah.
because I can tell you some of the things that he's presenting. It's almost like you must run up against this, Dr. Wischmeyer, the disbelief that fecal transplant can actually be useful, and I've seen reputable physicians out there that just say, this is complete garbage, it doesn't work, and then to see all that data there is just amazing.
It's compelling.
Do you think that when you mentioned about the increase in C. diff infections, I had a discussion at a conference on Friday with a physician on how he hardly ever sees GI bleeds in the outpatient department anymore. And so then I started wondering, is maybe the reason why you're seeing more C. diff infections, is it because there's a lot more PPIs being prescribed? Would that have anything to do with it?
That's absolutely true. I think that's a great question. And in fact, we just finished recently the Society of Critical Care Medicine PPI and antacid guidelines, looking at should we be routinely giving H2 blockers, PPIs, proton pump inhibitors to patients, and what does the data actually say about that? And what we found is that unless you have significant risk factors for bleeding, like you're an anticoagulant, you have septic shock, you are on some sort of platelet inhibitor, or you have DIC or some other very high risk for GI bleeding, there was not a benefit of routinely being on a PPI or an H2 blocker necessarily, especially a PPI on a routine basis. But what we did find was people who are on PPIs and even H2s had significant increases in both C. diff and pneumonia risk. So it's stunning to me in my ICU practice, every week. I would say at least 75% of my patients come in from home on a PPI. And I look for any and every reason to stop them, because not only do they increase your risk of infections, C. diff, and pneumonias, they also over time cause renal dysfunction. And so PPIs carry significant risks for people over time, and there's so much of the world that's on them. And clearly, if you have severe ulcer disease or really severe GI reflux, there's a benefit potentially to that because you're at risk. But I think a lot of people get put on them they have a little bit of stomach upset or a little bit of indigestion, and they've had it one time and they end up on them for months and years. And I think that's really devastating. And I think if you're on those and you really don't have sort of significant threats to having ulcer disease or really severe esophagitis or gastritis, that was something I would be looking for a way to get off of. Because like you said, I think one of the reasons we see less GI bleeds clearly is because we have such a preponderance of this, but that is leading to increased C. diff, increased pneumonias, and increased renal dysfunction over time in people. And those are big risks that I think are probably more common than even the GI bleeds we used to see.
Oh, Graham, we lost you there for a minute. I just want to get your we'll just wait to get your audio back here. Just one second. We'll try to unmute you in a moment and let you speak. But in the meantime, we'll just try to unmute Graham here. So many incredible insights during that presentation. If you have a question for Dr. Wischmeyer and you're watching from one of the platforms we're streaming on live right now, please feel free to ask your questions in the comments. We'd love to hear from you. We'd love to get your questions about gut health, about the microbiome, anything that Dr. Wischmeyer presented on today. I know there's a lot of interest in this topic, but we are presenting, and Dr. Wischmeyer presented everything from an evidence-based perspective. I also want to make sure that you know that this session, none of these sessions are sponsored by any industry or probiotics companies. We're not trying to push anything on you. We want just to make clear what the scientific evidence is for the use of probiotics and what it isn't, right? And so if you do have questions, we're also doing a book giveaway at the end of this, and any questions or comments will get you into that book giveaway. So Graham, your audio's back. And we were talking about proton pump inhibitors and concerns with the overprescribing, and often we call this polypharmacy. Well, this happens with polypharmacy sometimes, right?
Sure.
But Graham, go ahead.
Yeah, and that was completely my fault. I turned my mic off just so I could hear Dr. Wischmeyer talk. And there are actually online lots of great deprescribing sources out there for PPIs. Can you speak to the patients that have ileostomies, short bowel? Have there been a lot of studies done with probiotic effectiveness or any kind of results in these patients?
I have not seen very much research done in those areas. I will tell you, I don't have a colon and I'm short gut. I've taken a probiotic every day for many years now. But I don't know a lot of the literature. And one of the questions, of course, would be, without a colon to colonize, does it have the same effect? And likely it probably does still have an effect. I can tell you I worked as a COVID-19 ICU physician for all the years of COVID, and I was exposed to many, many patients who had it, and I never got it. Now, that's not really scientific per se, but I did have the belief that it would make a difference, and that was long before the vaccine was out that we were being exposed to many, many patients with it. But that said, I don't know that there's a lot of data in that area. But again, one of the things we do know about probiotics is there's a fair bit of data, especially in the GI inflammatory bowel disease world, that even a probiotic that's not alive, the cell wall components of that probiotic causes signaling at the intestinal cell signaling. So even if you take pieces of bacterial cell wall or the bacteria, people always say, oh, gosh, what if the probiotic I take dies in the stomach, or if it's a little older and the bacteria aren't alive anymore? Ideally, yes, we'd love to have the bacteria be alive and colonize the gut in a good way and reset the commensal sort of healthy bacteria of your gut. But even we know that heat-killed probiotics or pieces of the bacterial cell wall of commensal organisms can reduce inflammation, can positively modify a lot of aspects. And so it is likely that sort of throughout our gut, that the probiotics we take are signaling through our cells beneficial things. One of the things we found in some of our studies is, and I didn't present the basic science work that I've done with probiotics, but we did studies where we gave animals Pseudomonas pneumonia, where we injected Pseudomonas into the trachea. If we gave those animals a lactobacillus probiotic, LGG, before we gave them Pseudomonas, virtually none of them would get pneumonia, and their survival was very high. If we didn't give a probiotic a few hours before, 70 or 80% of them would die, and they'd get severe pneumonia. And one of the things we found was just the presence of the probiotic for a few hours in the gut improved the mucin barrier in the lung. So things we do in the gut have major effects in the lung. And this is one of the reasons we think it prevents viral and bacterial infections. We found that if you knock that mucin gene out, the gene that makes the mucin in the lung that protects the lung from infection, that this mucin in our lungs keeps bacteria from sticking and infecting, and viruses as well, that all that benefit of probiotic was lost. So we actually found a pathway that showed directly how bacteria given to the gut can very quickly change the expression of protective factors in our lungs, mucin and others, to prevent infection, even when we give lethal doses of a bacteria like Pseudomonas, which is a very severe pneumonia, to the animal. And so we found that very striking. These was some of our best publications in this area. And one of the reasons we like lactobacillus, other bacteria may do it as well, but this was the most effective of the ones we studied. We also found that in like ruptured appendicitis or in GI infection, giving a probiotic shortly before the onset of peritonitis or say ruptured appendicitis, significantly reduced mortality from that infectious source as well. And in fact, in both those models, some of the different T cell subsets that help ameliorate inflammation and prevent other organ injury was significantly modified by the presence of a probiotic versus not. So just the simple presence of a probiotic like a lactobacillus in the gut significantly modifies signaling in the lung, mucin production in the lung, and how our T cells respond and prevent infection. And so there are dramatic changes that happen very quickly from a probiotic administration that can be very protective against a whole range of infections and insults.
Fascinating. Graham, anything more to add, or?
Yeah, that part's interesting because, as a pharmacist, you're always thinking that you have to kind of keep giving it and keep giving it and keep giving it to have the small effect that it would have.
Yeah.
But if you have just even one dose, and it can have that quick effect in that period of time.
That's what we saw.
And I think, Lindsay, one thing that I've really learned, even from today, is that I think even in previous sessions, we've talked about the need for a probiotic. Yes or no, should you take it if you're a healthy person?
Right.
And really, we were kind of on the fence on that until today. Like, definitely after this, that I've changed my mind on that.
Yeah, one question that kind of came to mind was we have covered, the issue of or the need for, prebiotics like fiber, and the fact that in this industrialized society, a lot of us are eating a lot of processed foods. Our diets are not
Yeah.
great. You spoke in your presentation about the use of synbiotics in some of these studies, where you have the prebiotic and the probiotic combined, but you also spoke about, I think you even said yourself you take a probiotic every day. But is there any concern for people who might think that it would be good for them to take a probiotic daily if their diet is not a diet that is replete with good sources of fiber? Because ultimately these probiotics need to eat, and we don't want them eating, that mucin layer. So is there any concern with that, or yeah?
So yeah, it's a great question, Lindsay. I think I would advocate that the data is more compelling for the benefits of probiotic use by healthy people than in illness. And I think that's to some degree because we haven't done as many studies in ill people, and there's many different illnesses you could study probiotics in, and of course it's going to take time to accumulate literature in all these different disease states, whether it be cancer, transplant, surgery, critical illness. There's such a wealth of different illnesses you can study this in, and lots of different ways you probably need to administer them. So I would say the data that exists, if you look at the Cochrane analyses, the number of studies, the number of patient studies, the quality and strength of the studies, particularly with that Nature paper out there, is much more compelling that everybody should take a daily probiotic who's healthy than for any illness state, but except for maybe C. diff. I think C. diff would be the one exception I would give to that, where I think the data is very robust and very compelling. I think most of these studies were done with probiotics alone, so I think a probiotic alone can clearly benefit people. But that said, I think the very best studies are the ones that give them together. Like that Nature paper, for instance, I think is the gold standard. I mean, there is probably no better study in almost any area of medicine than that trial for that intervention. I would put that up against the studies that have been done in heart disease or cancer or anywhere else. I mean, that is a really well done study from a really, really strong research group. And I think that really showed. And when, it was interesting when I talked to him, the primary author of that, they chose that particular probiotic and that prebiotic because they did some studies ahead of time, actually based on some of our basic science studies in pneumonia, where they were looking for a probiotic-prebiotic combination that would lead to colonization and persistence of the probiotic species in the gut. And they found that a lactobacillus with a fructooligosaccharides seemed to lead to the best persistence of the probiotic colonizing and putting all the people back in New Orleans of the gut. And that was the reason they chose those two together. And I think given some data that I will hint at, that Mara Surbanescu, who is an amazingly talented anesthesia and critical care physician who we've brought to Duke from Johns Hopkins University, and she is now the only trained intensive care and surgical physician I know who can do her own microbiome analysis, like the computer scientists that do the work. So she's the only person in the field that I know that can do this. We have begun to look at the effects of fiber alone. And I will hint to you that in critically ill patients, giving fiber alone probably feeds the pathogens as much or more than it feeds the commensals, because there almost are no commensals. So the idea that you're going to give a prebiotic fiber to a depleted dysbiotic gut is probably not ideal. And that you should give them together, that you should give a commensal with that prebiotic, is probably the best route forward, much like that Nature paper did. But that said, I think if you're a healthy individual, taking a probiotic alone can have benefits, but the data would say taking them together would be better. I think the data is more robust in the well-done studies. And you saw there were 10,000 patients studied in that large symbiotic study. And so there is data out there for that, of course. So I would say that's the most compelling. But yes, I think a probiotic alone, there is very robust data from both Cochrane and some of the other large meta-analyses. Just say that a probiotic alone, most of whom were probably people not eating the best of diets, because that's most of the world, unfortunately, in the Western world, can still be beneficial. But if I was telling you the perfect thing to take, I would say take a combination.
Yeah, that's really interesting, and this is such a developing field, right, of interest and also just development, right? So thank you for answering that. We also inevitably had the question, which is a great question, and if you have questions for Dr. Wischmeyer, feel free to post them. We're doing our Q&A right now with Dr. Wischmeyer from Duke University. We had the question, what probiotic does the doctor take?
So
you don't have to answer. I'm just
I'm happy to answer. I take a Lactobacillus GG probiotic, and I will disclose that I take Culturelle. And there's one disclosure with that: they did provide the funding for our COVID study. They provided the product and some gift. It was not actually funding that was directed for any purpose. It was actually unrestricted gift funding. So it wasn't the classic sort of, we're going to give you money to study our probiotic. They gave us money to study COVID and the microbiome in general. And they said, just try to do good in the world. And they also provided us with the placebo and the probiotic. And the reason I think we use theirs in the hospital, most of the hospitals I've worked in have used that particular one, is because it's made and sealed in an anaerobic package. So they have very robust quality. Most of the large clinical studies that have been done in acute illness have been done with that brand and that probiotic strain. We also had robust packaging and quality control and manufacturing information to get 9D with it. So we were able to go to the FDA. They were able to provide really robust quality control information and manufacturing information to the FDA such that we can get 9D. And so again, I think a lot of us worry about, gosh, is the probiotic I'm taking actually what it says it is? Is it manufactured in a quality way? Is it still alive when I take it? And so I think the reason this particular brand, I suppose you would say, is used so commonly in the research trials, and the reason we use it, the reason I take it, is because we have so much information on its quality and manufacturing standard, and it has so much data in the literature for safety and then efficacy in many cases. That large study the NIH funded that showed a 50% reduction in ventilator-associated pneumonia, also cultural related Lactobacillus GG. So again, I think there are other quality probiotics that exist. I won't say that's the only one, but we also studied it because you can get it at Target, you can get it at the store, you can get it on Amazon. We wanted to study something that was inexpensive, widely available, that a developing nation person could take, or a person down the street in the US or Canada could take, and they could get it easily and access it easily. We didn't want some sort of esoteric, unique organism no one could get. That doesn't help much in a large global pandemic. So I think for a range of reasons, that was one of the reasons that I take that, and we chose that for our research. So good question.
That's
Do you
great.
find there's a difference between, sometimes I hear it's a good idea to switch up the probiotic you get from time to time rather than, is there any proof to that?
There may be, but one of the things that I learned, there's a group in Sweden that has maintained a massive microbiome database on diabetics, on type 1 diabetics, and they have a vast experience with people who take probiotics and sort of what their microbiome looks like and everything. And he made the point, and you heard me repeat it today, really any good commensal organism is going to outgrow a pathogen pretty much in every case. And that's why when you look at like that large JAMA meta-analysis and even the C.diff meta-analysis from Cochrane, there are multiple probiotic strains that have been used successfully against C. diff, against antibiotic-associated diarrhea, against venereal-associated pneumonia even. And many of them work. It isn't just necessarily one. And what I think that tells us is, in many cases, maintaining a good commensal, especially against an insult, let's say you've been on antibiotics, or you've been sick with big surgery, or been in ICU, or even had COVID, which also disturbs your microbiome. Taking many of the different commensals probably have somewhat similar benefit. I think I will say, given the Nature paper studied lactobacillus, I think there is some draw to the larger, high quality studies like that paper saying a lactobacillus may be uniquely beneficial. But I don't know that you could say that with any assuredness, or any sort of, it's not as though someone else has repeated a trial like that with another probiotic and said, oh, it does or doesn't work. So I think because of their ability to persist in the gut, and because they're good fermenters of short-chain fatty acids, and because they are persistent, and there's high quality manufacturers of them, lactobacillus have been studied. But again, I think there are other probiotics that could be of benefit. I don't know that switching them up makes a difference. I think some people also advocated take a multi-strain probiotic. I haven't seen data to say that that would be better than not taking a multi-strain probiotic. So I think those are excellent questions for research that needs to go on. To my knowledge, I don't think we have the kind of evidence we need to be able to answer that, but I think it's an outstanding question.
Yeah, I had the final question, Lindsay. I know that we've been on for a little while. You had mentioned about cell walls and inactive bacteria. So this is in the realm of, I guess, postbiotics is what we call it, right? So is there any study that just is looking into maybe just giving that?
Sure, and I will qualify this answer saying I don't know that literature as well. It's not something I've spent as much time on, but I do know that, especially in the IBD world, there is robust NIH funding at a number of institutions and a number of excellent scientists, labs, studying, like, essentially, I guess, what we call postbiotics, sort of these specific components of bacteria that seem to signal immune effects, especially immune signaling effects and other gut effects of these probiotics. So the data that I have seen is promising for sure. I don't know how much clinical data is, or maybe a fair bit that I don't know about, but I know that research is happening. I know there's a great deal of interest in it. It's a way sort of enriching or targeting specific targets with the different bacterial components. A little bit like, our COVID vaccine is a component of the virus and is very robust at signaling immunity. It's not the whole virus, obviously. The idea that you can take components that are particularly beneficial at signaling for what probiotics do and give them as individual components, I think has got a great deal of promise. And there are many very, very strong labs studying it. So I think there's definitely promise there. I don't know that I can cite the literature as I could say for probiotics or prebiotics.
Okay,
but
thanks.
It's a great question. Great question. And a big future.
Yeah.
Yeah.
That's great. We have one question here from a viewer. Now, this is a big question, and I know we're kind of short on time here. So, but I will just show it here. So, you talked a lot about diversity and the microbiome, and the knowledge I have is that we lose diversity as we age. So, can you just give us kind of your 2 cents on, how does aging affect this diversity?
We know, and we actually have a trial we're about to publish looking at this question, but we know that as you age, your microbiome becomes less diverse in general. Now, you can do things probably to improve that, but we know that as you age, it becomes less diverse. And one of the areas that perhaps I'll come back and talk about in the future is cognition and the microbiome. And there's a lot of data that is beginning to show that cognitive decline and delirium and dementia probably has got a significant relationship to the microbiome in the gut, but even more so in the mouth and the nose. In fact, in certain patients who've had significant dental work done as children, they've done these studies in Sweden and other places, people have greater dementia and sort of cognitive decline if there's been disruptions of potentially the microbiome that could expose potentially the brain or the sinuses to certain bacteria. And so we worked with the chair of my department, Joseph Matthew, who's a huge researcher in the areas of major cardiac operations, which are known to lead to cognitive decline in people who have them, and potentially dementia and other cognitive disorders. We did a large study with him. He did the study, and then we collected samples. And we have a fair bit of evidence now looking at the potential of what different microbial disruption and dysbiosis can do to cognition in those patients. And then we have another NIH-funded study that is just finished as well, where we collected microbiome samples from non-obese, healthy children. We collected them from cognitively impaired elderly people, and then the people that lived and cared for other elderly caregivers in the same home as the cognitively impaired people in some of our dementia and cognition Alzheimer's clinics. And so we're looking for microbially derived, we call them juvenile protective factors, basically, factors that don't exist in people who are aging and have cognitive disorders, that do exist in people with normal cognition and exist in children, because there are factors in bacterial families that exist in children that we lose in elderly folks. And our data is hinting, although I'm not going to share it all because we are about to publish it, but they're hinting that there are specific bacterial families and subsets and changes in their metabolites, right? The same bacteria might lead to a different metabolite in one person at a certain age versus another person at an older age. And there are clear metabolite changes that occur as we age that seem to have clear associations with dementia and Alzheimer's and cognitive dysfunction that may be targets to prevent or slow the progression of these aging-related, neurologic and cognition-related issues. And so this is a big question. It's one that we've actually got a fair bit of funding to study, and we're now actually finishing a lot of those studies. So we're really excited. I've actually seen the data from both studies, and there will be some we're excited about the findings, I will tell you. And these are studies that Mara Serbanescu, this incredible researcher who I really had the pleasure to mentor and train, who's going to probably far surpass anything I ever do, that I'm super excited for. She's been working on these for the last few months, and there's some cool, compelling data about how aging changes us, but also how people that are having cognition problems, dementia problems, Alzheimer's problems, how their microbiome has changed and how we might be able to do something about it, both maybe in prevention and treatment. And so the bottom line is it does change, and I think we should do all we can to maintain a healthy microbiome, especially as we age.
Yeah, that's excellent. Thank you so much for sharing
Yeah.
that. That is a huge question. But
Yeah.
obviously, yeah, a lot going on in that area. And Dr. Wischmeyer is coming back in a few weeks, and he's going to talk about the gut-brain axis. So for those that are interested, because I know a lot of you have expressed interest in this topic, and we've kind of been teasing you, saying that it's coming, but not letting you know who's speaking about it. So it's actually Dr. Wischmeyer is coming back, and it is a loaded topic. So in a future session, I think it's about 3 weeks from now, we'll be discussing this. Dr. Wischmeyer, anything else you kind of wanted to leave us with? I mean, I feel like I need to watch this presentation like 3 times to absorb it all. But.
Yeah, I think the compelling thing that I have found in the many years, I guess 20 years now, we've been doing this research, is some of the questions you actually asked, and there's some of the questions I think we all get asked a lot, whether pharmacy or physician or whatever our practice is, and I think one of them is it does appear to me that there's enough data now to say that it probably is reasonable that healthy people should routinely take a probiotic every day. Now, if you eat a lot of yogurt and cultured foods, sauerkraut and other fermented foods, can that give you the same benefit? It may and probably can. And so I would encourage you to eat foods that are fermented. I mean, clearly we know the people that live the longest, that have the longest lifespans, that live to the oldest ages, are in parts of the world where they eat a lot of probiotic fermented foods. And so this is not new data. We've known this a long time. And so clearly there appears to be a longevity and health benefit to taking in bacterially rich foods, which sort of goes against everything that we think about. We're like, oh, we don't want bacteria in our food. But I think short of that, we have such good data now to say there's sort of direct day-to-day benefits of taking a probiotic or prebiotic probiotic combination, symbiotic, in just preventing the illnesses and complications that plague our lives. The COVID infections, the influenza infections, the respiratory tract infections that keep us sick and make our kids sick and keep our kids out of school and us out of work. Those appear to be significantly preventable, and the severity of them will be significantly less if each of us was to do that. And I think just based on that alone, much less its potential other benefits on cognition and on other parts of our health, I think is a robust reason. And then I think the data is obviously growing for specific illness states. And I think the idea that we'll all be freezing our stool soon, short of us undergoing a bone marrow transplant or organ transplant or other major surgery in our lives, the idea that we might be getting our own stool back, because clearly getting your own stool back is much better than getting someone else's stool back, where there's, of course, always a small risk of autoimmunity and other complications. Probably has also got a future, and the NIH is actively funding some trials like that now. So maybe stock some things back in your freezer, but also consider taking a probiotic symbiotic now. And I think that's the message I would leave you with.
That's wonderful. Yeah, thank you so much. I mean, there's just so much research going on, right?
Yeah, there really is.
We really don't hear a lot about it until we speak with someone like you. So thank you for presenting that. And again, Dr. Wischmeyer will be back with us in a few weeks to speak about the gut-brain axis. So thank you so much for joining us. This has been so enlightening. I do want to do our book giveaway. The winner of our book, and actually this Myra Churchman is from Quesnel, British Columbia, Canada. She had joined us and asked a few questions. And Myra, it's your lucky day because you're not only going to get a copy of Gut Logic, Graham has also in this session said that you're also going to get a copy of his first book, which is Healthy Logic. These are both evidence-based resources about health, and I've read them both, contributed to one. And so Myra, if you can email me at friendlypharmacy5.com, we will send that out to you quickly. So thank you so much for that, Graham. And then I just want to share with everyone our upcoming sessions. Next week, Dragana Skokovic-Sunjic, the author of the Clinical Guide to Probiotics in Canada and the US, will be with us to talk about probiotics and gut health. Following that, she's also going to do a special session on May 11th, and where she's going to be speaking specifically about probiotics and women's health. And again, I spoke about Dr. Wischmeyer, who will be coming on again to speak about the gut-brain connection, which we're all interested in. And then we'll also be discussing a bit more about how to improve gut health and microbiome function. So those sessions are still coming up. The sessions in red are posted and are available for viewing. So thank you all so much for joining us today. Thank you, Dr. Wischmeyer, for giving so generously of your time and your knowledge here. Really appreciated it. Graham, any further thoughts?
Just such a great lecture to listen to. I would definitely, like you said, I would listen to that 3 or 4 more times just to get it all in. Thank you, Dr. Wischmeyer, for being here.
You're welcome.
Appreciate it.
Yeah, and
My
thanks
pleasure.
for speaking to the evidence, because there's just a lot of out there that is not based on evidence, and you've just done an incredible job of just really showing us the evidence today, and that's the whole point of what we're doing here. So thank you so much.
You're very welcome. Thanks for having me. This wonderful forum. I'm really thrilled you created it, and I think you're doing the world a great service, because I think you're right. There's a lot of misconceptions and lack of evidence out there, and I think this gives us a chance to really show people what the real evidence and the science is, and really get them the best information.
Absolutely. Thank you so much, everyone. Take care and stay healthy, and we'll see you in the next session next Saturday, May 4th.